Great attendance. So today I want to talk about advancing the understanding of HAP40's function as well as its role in Huntington's disease. So first of all, I want to start with a short introduction. So as most of you likely know, HAP40 forms a pretty stable complex with the Huntington protein. And as it was shown in the brain tissue, Huntington is the most abundant interactor of or HAP40 is the most abundant interactor of Huntington in the murine brain. And as we later showed, the two proteins are also highly evolutionarily conserved, and also their interaction is conserved in all metazoans. So for example, we could show that the interaction between Huntington and HAP40 takes place in human, mice, zebrafish, and it has been also shown in Drosophila. Furthermore, bioinformatical analysis by our group revealed that the two proteins have co-evolved, so meaning that when one amino acid change occurred in one of the proteins, there has been a complementary change in the other protein. The interaction is mostly mediated by large electrostatic and hydrophobic interaction surfaces, but the function of HAP40 as well as of the Huntington HAP40 complex remains unclear until now. What is known so far for the involvement of HAP40 and also for the interplay on a functional level between HAP40 and Huntington is that the HAP40 protein levels strongly depend on the protein levels of the Huntington proteins. So for example, we and others have shown that the levels correlate in diverse tissues. So as you can see on the side of the slide, there is close to perfect correlation between the protein levels of Huntington and HAP40 in fibroblasts, lymphoblasts, in the striatum, and in the cortex. Moreover, by doing Western blot analysis, we showed that by upregulating the Huntington expression, you also see increased HAP40 protein levels, whereas on the other side, if you knock down Huntington, you also see an reduced HAP40 levels. On the contrary, if you alter the HAP40 levels, there is no clear detectable change to the HAP40 levels in our experiments. So, moreover, we then wanted to investigate what is the mechanism behind this dependence on the protein levels. Therefore, we first started with performing qPCR assays and could not observe any alterations in the HAP40 mRNA levels up in Huntington overexpression, whereas another group, so the group of Su et al., could see some small variations in the HAP40 mRNA levels, but they were opposite to the changes in the protein levels. So meaning that when we increase Huntington levels, although we have elevated HAP40 levels, we have slightly reduced mRNA levels, for example. And therefore, later on, we could also show that the intracellular stability of HAP40 directly depends on Huntington protein levels, as the stability of the HAP40 proteins inside cells increases when the Huntington protein levels are increased. So similarly, we also performed Western blot analysis in various tissues of Huntington disease patients, and also analyzed data from mouse models, and we could see that the Huntington as well as the HAP40 protein levels are reduced in all those tissues. So for example, as you can see on the slide, there's a reduction in fibroblasts, lymphoblasts, as well as in the striatum of a Huntington knock-in mouse model. So therefore, the questions arise whether the loss of the Huntington HAP40 complex or the functional interplay between the two proteins contributes to the HD pathomechanism. As I've already mentioned, there is a strong co-evolution and also evolutionary conservation of the two proteins and especially of their interaction, which corroborates the hypothesis that those two proteins together fulfill an important function in the cellular metabolism. Moreover, as explained previously, the protein levels of the two proteins are highly correlated, and the levels of HAP40 directly depend on the levels of the Huntington protein. And as shown in the past, the Huntington knockout is embryonically lethal in mice, and also a full knockout of the HAP40 protein is also embryonically lethal, as it has been shown in a recent conference in Boston. So therefore, the main question currently is, what is the most important HD relevant function of the Huntington HAP40 complex? So you can see here the structure of the Huntington HAP40 complex, and it's a really huge protein where we believe that it is an ideal interaction hub. And that is also corroborated by the fact that more than 1,000 Huntington interaction partners have already been reported, but the main problem here is to identify which are the most important interactors of the complex out of this vast number. And especially also what are the direct interactors of the Huntington HAP40 complex. Therefore, we proceeded with studying the interactome of HAP40 to discover HD relevant functions. So for example, as we believe that HAP40 is central to the biology of Huntington due to the abundance of the interaction and the evolutionary history, we started with studying the interactome of the HAP40 protein, as this has been never done before. Further, one advantage of studying HAP40 at the beginning instead of the big complex is that the surface of the HAP40 protein is smaller than the Huntington HAP40 protein, which most likely leads to a lower number of expected of interactors that can be then studied more easily functionally. Also, as HAP40 is the main partner of the Huntington protein, and also as both levels are reduced in tissues of HD patients and mouse models, this will most likely lead to the identification of HD relevant functions as well. So here I want to show you some early data on our interatomic analysis. And for our interactome studies, we decided on following a dual APMS approach, meaning that on the one side, we wanted to quantify in vivo interactions by expressing HAP40 protein with a tag inside of human cells and then pulling out and fishing for Huntington interactors, HAP40 interactors, sorry. But on the other side, also by performing an in vitro interaction assays where we purified HAP40 and immobilized it on beads to identify interactors of HAP40 using a second method. And as you can see in the Venn diagram, there is a relatively big overlap between the two approaches, although we can also identify some proteins that are present in only one of those approaches, but those are mostly proteins with a low enrichment in those assays. So most of the major identified interactors are present using both approaches. And on the other side of the slide, you can see here the graphical representation of the median log2 fold change on the x-axis and the logarithm of the base factor, which is kind of equivalent to a p-value in some sense, which we use for a measure of statistical significance. And just to show that some of those proteins have a relatively strong enrichment, and unsurprisingly, the most enriched protein in the in vivo interaction assays is Huntington protein, with a median log2 fold change of about 7. Next, I want to talk a little bit about our statistical approach in analyzing this data, as we decided to use a different approach than it is mostly used. So I will not go into detail on the exact statistical background of those models. So we decided on using a hierarchical Bayesian model, which has more or less the advantage that it captures the uncertainty. So we can capture the uncertainty within each experiment, and this is then also propagated to the final experiment. So thereby, instead of just having a single point estimate, we end up with having distributions of the enrichment values, which give us on one hand, information on the certainty we have in those log2 fold change, which later helps with discriminating between highly went and low went hits, because there may be proteins which have a pretty broad distribution. And we also may end up with proteins that are enriched only in one of the experiments or two of the experiments and not in all of the experiments. And by really comparing the posterior distributions, we can get much more information out of the statistical analysis. Additionally, this method can help us with differentiating between direct and indirect interactors of the protein. As you can see here on the side, there's a graphical illustration of a theoretically possible complex between the HAP40 protein and 4 other proteins. So you can see here, for example, that the protein P1 is directly binding to the HAP40 protein, whereas proteins P2 and P3 would then bind to this P1, to protein 1, and then there is a third protein, P4, which is then interacting with proteins 2 and 3. And then by plotting the posterior distributions of the enrichment value on, in a histogram and density plot, you can see that, for example, the direct interactor has the highest fold change and enrichment of all the interactors, and due to the fact that we have those posterior distributions, we can also calculate statistics based on that to really say that this is a significant difference. So next, I want to go a little bit into detail of our current results. So we are relatively early in investigating the interatom of HAP40 so far, but I want to show you the results of a GO-term enrichment analysis. So the most enriched biological process and also cellular localization of the interactors that we found for HAP40 in this dual APMS approach are the mitochondria. So there we have a relatively strong and statistically significant enrichment of those mitochondrial proteins, which is with an enrichment factor of about 5.5, meaning that there are 5.5 times more mitochondrial proteins that can be expected based on a random sample, which may imply that HAP40 has some function in the mitochondrial biology. And on the other hand, we could also identify proteins that have been previously shown to be involved in neurodegenerative disorders, especially proteins that have been connected, with Huntington's disease using this GO-term enrichment. So we could identify a lot of proteins that were previously associated with HD in the interactome of HAP40, which also corroborates our hypothesis that HAP40 may play a role in the function, in the pathology of HD. So as a first functional assay, we analyzed the cell proliferation of HAP40 knockdown cells using 3 different starting conditions, and you can see that in all cases, the cell growth of cells with an HAP40 knockdown was diminished in all experiments, which raises the question whether the reduced cell proliferation may indicate a mitochondrial dysfunction, as the function of mitochondria is important to the cell proliferation. So as a conclusion and short outlook on our ongoing work, I want to sum up by saying that our dual APMS approach, combined with the statistical process, allowed the identification of promising HAP40 interactor candidates, and we could show a strong enrichment of mitochondrial proteins and partly also of nuclear proteins among the identified HAP40 interactors. Moreover, functionally, we could show a reduced cell proliferation up in HAP40 knockdown, and will now go on by confirming some of the identified interactors using complementary approaches, and then also studying the role of HAP40 in mitochondria as well as the role of these interactors in HD. So finally, I want to thank all the people involved in this project and also especially the EHDN for funding this project and you for your attention.
Thank you very much. Are there any questions from the audience? Maybe while they're thinking about it, a very naive question or two. You mentioned that the relationship in overexpressing Huntington and HAP40 is that they go up. If you knock them down, they go down. You have tried this in transgenic and overexpression models. Have you tried this in treated cells with ASOs or something that's more in a therapeutic line? So if you try to treat and bring down the levels of Huntington, will the levels of HAP40 also go down?
So we haven't done this ourselves, but the other group I mentioned from Su et al., they looked into, they treated the cells with siRNA to also reduce Huntington levels, and there they could also see reduced HAP40 levels. So it's most likely that as soon as you lower the Huntington levels, you have lowered HAP40 levels, due to the dependence of this protein stability.
What you see with your pull down.
So the problem with immune fluorescence or localization studies with HAP40 is a little bit that the quality of the antibodies is mostly not sufficient. So a lot of those antibodies are not available anymore, so we are also generating our own antibodies now. And a lot of the HAP40 protein, as previously shown and also by us, is also localized inside the cell nucleus. But so far, we're not succeeded in staining the Huntington HAP40 complex with any HAP40 antibodies. So also in cell staining, we cannot see a clear co-localization or strong co-localization between the two proteins. So it may be that the epitope that is detected by the antibody is also masked in a complex. So we are continuing with that, and also by doing in silico analysis of already available data on the proteome of mitochondria, HAP40 is not detected in there. So it seems to be that it's rather probably on the surface than really inside of the mitochondria. So for the affinity pull down, we actually used a tagged version of HAP40 and used an antibody against the tag. But if you look at overexpressed HAP40, it's more in the nucleus than in anywhere else. Yeah.
If there is no more question, thank you very much for your presentation. We move to the second speaker, the regional speaker as this morning, so Karine Merienne from the University of Strasbourg, and she will speak about epigenetic dysregulation in Huntington disease. Karine, the floor is yours.
So thank you, Nicole. So first I would like to thank the organizer for the invitation, and actually I'm very happy to share our data here in Strasbourg. So I will talk about epigenetics in Huntington. As you know, selective neuronal population degenerate in Huntington disease, particularly spiny projection neuron in the striatum, both direct and indirect SPN, while other neurons in the striatum, like interneurons, are relatively spared. Similarly, in the cortex, you have also selective neuronal degeneration. For instance, layer 5A pyramidal neurons are highly vulnerable, while other neurons in this layer, like the beta cells, appear quite resilient. So the mechanism that underlies this selective neuronal degeneration is not fully understood. However, since the first GWAS study, almost 10 years ago, which revealed that mismatch repair genes are critical genetic modifiers of the disease, we now have accumulated strong evidence that somatic CAG expansion is a major driver of neuronal vulnerability in HD. Moreover, recent study, particularly mouse studies generated by the group of William Young, actually showed that the mechanism is transcription dependent and requires extensive transcriptional dysregulation. So if you block somatic CAG expansion, this suppresses not only neuropathological features, but also this suppresses transcriptional dysregulation. And recent human data now support this idea, and I guess that you will hear more about that tomorrow by Nat Hines and Bob Hansecker. So in fact, both mouse and human study indicate that transcriptional dysregulation occurs when the number of CG repeats is long enough and above a threshold of about 150 CG. So it's much higher than the genetic threshold of 35-40 CG. And this transcription-related threshold is reached through somatic expansion, which is faster in vulnerable neurons. So one last important thing is that the dysregulated genes in those vulnerable neurons, they display specific signature. Particularly, the downregulated genes are enriched in identity genes. For instance, in spiny projection neurons, they are enriched in genes that define the identity of the spiny projection neurons. And the upregulated genes also have a specific signature. They are enriched in developmental genes that are normally repressed in mature neurons. So the question is how long CAG repeat and somatic CAG expansion drive transcriptional dysregulation. And I would like to show you data that suggests that epigenetic mechanism is a critical player. So very briefly, epigenetic mechanism modulate the structure of chromatin and control transcription through three different mechanisms: DNA methylation, non-coding RNA processes, and histone post-translational modification. And in the lab, we focus essentially on histone post-translational modification, including histone acetylation, such as H3K27 acetylation, which is associated with open and transcriptionally active chromatin. And also we are interested in repressive marks, such as H3K27 trimethylation, promoting repressive transcriptional repression. So epigenetic mechanisms are critical during development and cellular differentiation, where they drive the acquisition of a cellular identity through transcriptional reprogramming. So in progenitor cells or in differentiating cells, proliferation-related and developmental genes will be on, which is associated with high level of K27 acetylation and low H3K27 trimethylation, while cell-type specific genes will be off and will have a high level of H3K27 trimethylation and low level of H3K27 acetylation. In contrast, in differentiated cells, the situation is completely opposite. For instance, in mature neurons, you will have transcriptional activation of neuronal specific genes, and this will be associated with high level of H3K27 acetylation, and in contrast, you will have repression of proliferation-related and developmental genes, and they will show high level of H3K27 trimethylation. So those two histone modifications are really cell-type specific marker. They are marker of cellular identity. Last, epigenetic mechanisms are critical during cellular aging. And in fact, the recent paper by the group of David Sinclair showed that cellular aging is caused by a loss of epigenetic information that induce a loss of cellular identity. So briefly, what they found is that during aging, you will have a loss of H3K27 acetylation and a gain of H3K27 trimethylation at cellular identity genes, which will lead to their transcriptional downregulation. And you will have a derepression of developmental genes due to a loss of H3K27 trimethylation and a gain of H3K27 of histone acetylation. And they call this loss of cellular identity due to epigenetic aging X differentiation. So the question is, what's going on in Huntington disease at the epigenetic level? So to address this question, we perform epigenomic approaches. Particularly, we did some ChIP-seq experiment, which is chromatin immunoprecipitation coupled to high-throughput sequencing, which generate epigenomic map at genome-wide level. So I'm going to briefly summarize what we published so far. So initially, we generated ChIP-seq data using bulk striatal tissues on R61 transgenic mice, as well as our Q140 knock-in mice at different pathological stages, early and late stages. We also generated data in the striatum of HD patients, and we targeted different histone modification, particularly H3K27 acetylation. And so to make a long story short, we made two major conclusions. So first, we found that there was an extensive epigenetic dysregulation in the striatum of HD mice and patients. And more specifically, we found that there was a depletion in H3K27 acetylation at the broad regulatory region, so-called superenhancer, that regulate the genes that define the cellular identity genes in the striatum, so striatal neuron identity genes. And we found that this signature was conserved across HD mice and also in HD patients, that it establishes early and it is progressive in mice, and that it correlates with transcriptional downregulation of the striatal neuron identity genes. So this is to illustrate the idea, focusing on one of such striatal identity gene, PD10A, which is highly expressed in the striatum, and more specifically in spiny projection neurons. It is also strongly downregulated in HD, both in mice and also in humans. And this is to show you that H3K27 acetylation is strongly decreased at the two regulatory regions, E1 and E2, that are upstream to the promoter of PD10A. Moreover, we perform also, we investigated the 3D chromatin architecture at PD10A locus, so doing chromosome configuration capture-based approach that is called 4C-seq. And so what you see here is an interaction maps that show that the interaction between PD10A promoter and the two upstream regulatory region is weakened in R61 striatum as compared to wild type. We did similar experiments in knock-in mice and found similar results. So this is suggesting that downregulation, transcriptional downregulation of PD10A results not only from loss of H3K27 acetylation, but also from disruption of a chromatin architecture, 3D chromatin architecture. So the second major conclusion we made is the following. Based on our ChIP-seq data generated on the striatum of Q140 and control mice of 2 and 6 months, so we found that the loss of H3K27 acetylation at striatal neuron identity gene was accelerated in HD mice. And this is illustrated with this graph showing that there is a positive correlation between H3K27 acetylation changes in Q140 versus wild-type mice and the changes between wild-type mice of 6 and 2 months. So together, our data indicate that there is an accelerated loss of H3K27 acetylation at striatal neuron identity, suggesting that there might be an acceleration of epigenetic aging in striatal neurons. However, to prove this, we also needed to show that there is an accelerated epigenetic derepression of developmental genes, which is the second major feature of epigenetic aging. So one limitation to our previous experiment is that they were performed using bulk striatal tissue, which prevented precise cell type specific analysis, and particularly the analysis of developmental gene in striatal neurons. So to overcome this limitation, we generated a new series of epigenomic experiments using R61 and Q140 mice, as well as the controls. But this time, we performed the experiment on purified neuronal nuclei and non-neuronal nuclei, using FANS for fluorescently activated nuclear sorting, and we used NEN as a neuronal marker. So we generated FANS ChIP-seq and also FANS cut-and-tag data. So cut-and-tag data is a more recent approach which provides similar information as compared to ChIP-seq, and targeted again different histone modification, including H3K27 acetylation and K27 trimethylation. So I'm going to start with the FANS ChIP-seq data we generated in R61 mice. So we performed differential analysis, which showed that hundreds to thousands of genomic regions were differentially enriched in H3K27 acetylation and H3K27 trimethylation, specifically in the NAN+ population, so in neurons, while you can see that in the NAN- population, very few changes, we could observe very few changes. So now, due to a short time, I'm going to focus on the regions that are depleted, the genomic region that show depletion in H3K27 trimethylation in R6 versus wild-type striatal neurons. So we perform a functional enrichment analysis, which show that the genes that are associated with this loss of H3K27 trimethylation in striatal neurons were enriched in developmental genes. We completed the analysis interrogating the HD signature database and found that those genes that show loss of H3K27 trimethylation were also enriched in genes that are upregulated in HD striatal neurons. They also display, they were also regulated by the polycomb repressive complex, which was confirmed by a transcription factor prediction analysis using the database G. So to summarize, gene depleted in H3K27 trimethylation in striatal neurons of R61 mice present a developmental signature, are controlled by the polycomb repressive complex, which might be expected for developmental genes that are repressed, and are transcriptionally upregulated in HD striatal neurons. So this is to illustrate our data. This is a snapshot of our ChIP-seq data focusing on one such developmental gene, which is 1CUT1. So you can see that H3K27 trimethylation is, I don't know if it's working, yes, is depleted in the 9+ population in our 6-1 mice as compared to wild type, and that this is associated with a gain in histone acetylation, including H3K27 acetylation, K9 acetylation, and K18 acetylation. And the changes are quite specific to the N+ population, since it is not really the case in N- cells. So this is associated with transcriptional upregulation of 1CUT1, and particularly in spiny projection neurons. So this is true in mice, but we can also observe this upregulation in human data. So, okay, these data indicate that there is an epigenetic derepression of developmental genes in striatal neurons of HD mice, but now the question is whether it is really accelerated. And so to answer that question, we perform temporal analysis, and this time we use the Q140 knock-in mice, which represent a slowly progressing HD mouse model as compared to the R61 transgenic, so it's more suitable for temporal analysis. We analyze the mice at 2, 6, and 10 months, corresponding to prodromal, early, and advanced stages for Q140 model. And we generated fans cut and tag data focusing on the N+ population. And this is how our data look like. Again, I'm focusing on 1CUT1 gene. You can see that H3K27 acetylation appears to be increased over time in Q140 striatal neurons, while H3K27 trimethylation appears to be on the opposite, decrease over time in Q140 knock-in mice. So we actually quantified this, computing a euchromatin index, which is based on both H3K27 acetylation and K27 trimethylation signal. And this is really showing that there is an acceleration of the epigenetic derepression of 1CUT1 over time in Q140 striatal neurons. So this is to show that this is also associated with a progressive upregulation of 1CUT1 in knock-in mice. So we could actually generalize this data, computing this euchromatin index on the developmental genes that we found derepressed in R61 mice. And again, this is showing that there is an acceleration of the epigenetic derepression of developmental genes over time in Q140 striatal neurons as compared to wild type. Finally, we could use this euchromatin score to calculate the epigenetic age of Q140 striatal neurons. And actually, this graph is showing that the striatal neurons of Q140 knock-in mice of 6 months are older by 3 months, 9 months. And this is consistent with earlier study by Steve Horvath, the group of William Young, using the classical epigenetic clock based on DNA methylation, and showing that there was an acceleration of epigenetic age in affected tissue from HD patient. So this is showing together that there is an accelerated epigenetic derepression of developmental genes, and together with the progressive loss of euchromatin feature as striatal neuron identity gene, Our data support the idea that there is an acceleration of epigenetic aging in vulnerable neurons in HD, which probably triggers transcriptional dysregulation, the loss of neuronal identity, and subsequent neuronal dysfunction and death. So I didn't have time to tell you the full story. Please, if you want to hear more about that, go and see the poster by Baptiste Brûlé. And if you want to hear additional epigenetic story, you can also go and see the posters by Gilles Scuto and Noémie Penaud, which are PhD students in the lab. And so finally, I would like to thank the people implicated in the work. I mentioned Baptiste, but I also like to highlight the work of Rafael Alcalavida, a former postdoc in the lab, the other team members, our collaborators, funding sources. Thank you.
Thank you very much. The discussion is open. Any question from the audience? Don't be shy. Yes.
Really interesting talk, Karine. So I wonder, could you speculate what's the sort of connection between the mutant Huntington DNA, RNA, protein, and these very interesting and specific epigenetic changes?
So, well, that's the question we don't know yet. So the idea is that probably we need to reach this 150 CG repeat, this threshold, and then maybe we don't know how it works, but it seems that maybe it could be also require the presence of nuclear aggregates. But I guess you're also looking at this and. So, but we don't know, and maybe it could sequester some transcription factor, but we don't know how it could influence the chromatin. That's the question we still have to answer.
Very nice talk. I'm wondering if you have analyzed the topologically associated domains, if there is any change in the chromatin architecture in the nucleus, if the location of the heterochromatin changes or the active genes are located differently in the nucleus. Yeah.
So we did not investigate the 3D chromatin architecture genome-wide, so the experiment we did were focused on selective loci, but that would be interesting to do that, yeah.
Thank you very much for this fantastic talk. I'm a bit confused, you say about de-repression of developmental genes, so the brain gets to the more towards like proliferative thing and regenerates. I'm a bit confused.
So I don't think that you go back to proliferation, but it's a kind, indeed, of reversal that you do see during physiological cellular aging. That's what is mentioned by, what is published by the group of David Sinclair, that you lose epigenetic information that leads to a loss of cellular identity. It's not a complete loss, but you go back a little bit. Yeah.
Okay. So the senescent, so the dead or dysfunctional neurons die to make a space for stem cells then to produce neurons. Perhaps it's like this.
So the question is whether it could be a kind of compensatory response?
Yeah.
Well, we don't know.
Well, if there's no more questions from the floor, we'll thank Karine again for a wonderful talk. And it's my pleasure to introduce my co-chair, former colleague, and Professor Nicole Deglon from Lausanne University Hospital, who will be talking about genetic engineering to rescue Huntington's disease. The floor is yours.
So first of all, I would like also to thank the organizer for the kind invitation. So I would like to talk about genetic engineering, and in particular, I would say genome editing and how these new methodologies have somehow created a revolution in the field, which has been acknowledged with the Nobel Prize, which was awarded to Jennifer Doudna and Emmanuel Carpentier in 2020 for the discovery of the CRISPR-Cas9 platform. And this platform is unique in the sense that it recognized the DNA with an RNA molecule compared to the previous platform, which were necessitating the genetic engineering of protein to recognize a unique DNA fragment in the genome. And once this RNA is bound to the Cas9 protein, the nuclease, it then induces a double-strand break in the DNA. And what is this double-strand break inducing? It's inducing an inactivation because it introduces a premature stop codon in the gene of interest. So that's the basic initial function, but this CRISPR-Cas9 is really a Swiss Army knife with many options, and you can add modules to the Cas9 and therefore create additional function to this platform. You can, for example, repress or activate transcription. You can modify or even introduce new information into the genome, so write or modify the genome. You can also repair the genome. And why all these modules and this function? Because the human genetic disorder are very diverse with various mutations, and we need all these modules to be able to tackle most of these disorders. So today I will talk about what we can do with gene editing for polyglutamine disorder. And I would say there are three big categories. The first one is based on the original technology, so to inactivate either allele-specific inactivation or global, so both the wild type and the mutant allele. So that's the first strategy. Then we can also consider repressing the mutant gene. This has been done with zinc finger or CRISPR-I. And finally, we can also consider to induce CAG contraction, and this has been also proposed with these three strategies. Because of time constraint, I will focus on the study we have performed in the lab, and in particular, using the first strategy to inactivate. And we have published in 2017, a study showing that we can indeed very nicely and efficiently inactivate the Huntington gene, both the mutant and the wild type, in the striatum of mice. But of course, this opened the question, what about the function of the wild type gene and the long-term consequences of inactivating a wild type gene? And in the first part of my presentation, I will show you some of the results why we have tried to design an experiment to investigate this aspect in adult mice. We have also more recently started to work on a second pathology, which is Cas3, which is also a polyglutamine disorder, which has the advantage that the gene is very small, and we can therefore develop a very close strategy. We can also inactivate, so ablate, as I call it, both wild type and mutant gene, but because the Cas3 gene is very small, we can also replace and add a new copy, CRISPR-resistant, of this Cas3 in our animals to maintain the function of the wild-type gene. So let's move to the first part of my presentation. So the idea was really to revisit a little bit the impact of Huntington inactivation in the context of all the Huntington lowering program which are ongoing, and in particular the long-term consequence in adult brain, and for that we decided to work with wild-type mice and completely inactivate the wild-type Huntington in adults. And what we know is that the consequences, and there are accumulating evidence in the literature indicating that the consequences of Huntington inactivation are age-dependent, so during development, definitely it's not a good idea. In adults, the outputs are truly region and cell type specific. And we have certainly to go a little bit deeper, and a case-by-case analysis is needed. So to do that, we decided to do an experiment where we inject viruses, AAV vectors, with our gene editing system that we call Kamikaze, doesn't matter too much for today. And we are injecting in the striatum, but because of retrograde transport properties, the virus is going in the old projecting area. And we analyzed these mice over 11 months. And what we saw first in terms of behavior, we did motor behavior and anxiety-like behavior, is that there is absolutely nothing, no difference between the treated and untreated animals. So no impact, at least based on the test we used, of inactivating the wild-type Huntington in these animals. Then we look at the neuropathology, and in particular, all the transduced cells in the brain express GFP reporter gene, and you can see that there is no difference between control and treated again. And similarly, we have no astrogliosis or microglial reaction. And furthermore, the DARP32, which is a striatal marker, is unaffected in these animals, so which suggests that at least there is no major toxic effect or cell death occurring in these animals. So we pursued the analysis, and we then verified that indeed we have edited the Huntington in these animals. And for that, we took punches from the striatum and from the cortex, and we analyzed the editing efficiency. And as you can see here, we reach 53% editing efficiency. And if we consider that we only transduce neuron with the vector we have used, it means that we have targeted most of the medium spiny neurons in the striatum of these animals. In the cortex, we reach a little bit lower efficiency, and this is because of the somatotopy and the connectivity between the striatum and the cortex. We could not transduce all neurons in the cortex for sure, and we therefore only reach a level which are between 5 and 15%, depending on the location in the cortex. So indeed, we inactivate the wild-type Huntington in these animals. And this corresponds to 77% of the B allelic inactivation of the SPN and 16% in the cortical projecting neurons. So then we verified that indeed by doing so we have an impact on the protein itself. So by immunohistochemistry you can see that we have a nice drop of the Huntington in the treated samples compared to the control. And this is also true by capillary Western blot, where we have a nice drop of 50%. It's not 100%, of course, because you have also axons coming from the cortex and other regions which are still expressing the Huntington. But we have the expected drop of Huntington protein in the samples. So then in the last experiment, we did a single nuclei sequencing to see what are the functional consequences of this inactivation. So we took 4 mice per group. We did nuclei isolation, 10x single nuclei RNA sequencing. And out of this, after quality control and elimination of the poor nuclei, we end up with 100,000 nuclei, which is quite a massive amount of nuclei to be sure that we were indeed highly statistically significant. We did the cell type assignment based on this publication, and without any doubt, we identified the major cell types from the striatum, and the proportion fits extremely well with the previously published analysis. We have just 20 times more nuclei per group in our samples, so statistical power which is much higher. So then we use a new bioinformatic pipeline which is called Augur, which is specifically for single-cell analysis to help us identify and prioritize the cells which respond more to the inactivation. So which cell type is more affected by the inactivation. And this is important because in the striatum there is the majority of the cells are medium spiny neurons. And therefore, if you do the classical analysis, because of statistics, you will have more cluster and differentially expressed gene in spiny neurons. And what this lollipop plot is showing you is that here 0,5, it's random, so no significant change, and 1 would be an absolute change. And what you see is with all the cells are barely above 0,5, and in the pink area, these are the cells we have transduced. So definitely, it suggests that there is very limited. In a typical experiment using this platform, this Augur pipeline, dysregulation should induce value between 0.07 to 0.8. So we are truly below that level. We did also the classical differential expression analysis, and as you can see here, we have very few genes. Look at the number here, below 100 genes, whereas if you do the same with HD transgenic mice, you end up with 3,500 genes, which are dysregulated. So we are 30-fold lower than what we observe in HD mice. The price to pay for this very low number is that when we then use classical pathway analysis and term to try to figure out what are the consequences of this change. None of them end up statistically significant because we have very few genes belonging to each platform, to each pathway. So we are still struggling and trying to figure out to see what is the functional meaning of this dysregulated gene. But at this stage, we could say that long-term and efficient inactivation is obtained in wild-type mice, and it's quite well tolerated. It suggests that the continuous expression throughout life might not be essential to preserve neuronal survival and homeostasis. But what is even more important, I think, is that we truly need to have a case-by-case analysis, and we cannot draw conclusion from that experiment on other brain region. We have really to analyze the experimental model, the deliverable model. Partial inactivation with ASO is completely different from gene editing, where there is a complete inactivation. Targeting the striatum, the thalamus, and so on, or targeting the neurons of the astrocyte will lead to complete different outcome, and we have to go a little bit deeper in our conclusion and perform truly a case-by-case analysis in each cases. So that's for the HD. Let's move in the few minutes which are left to the SCA3. So in that case, it's the ataxin-3, which is with a CAG expansion, and it's a D-ubiquitinase. What we know is that ataxin-3 knockout has no phenotype, so it suggests that the function might be redundant. On top of that, there is a paralog in human, SCA3L, which intriguingly has no intron and no CAG expansion because the CAG are interrupted. And this is probably due to a retrotransposition, but it's occurs in primate, which means that mice do not have this paralog, but humans do have. And on top of that, ataxin-3 is a small gene, so we can put the cDNA, which is 10 times smaller than Huntington, in an AAV to do not only an ablate, but also a replace with a copy of the wild-type gene, which is CRISPR resistant. And that's what we did. So in that case, we are not targeting the striatum, but the cerebellum, which is another challenge because it's a very large structure and you need to deliver your virus in a very large structure, but we managed to do so. And we first tested 2 candidates in vitro, and we select the guide 2 for the in vivo study, and we show that indeed in the cerebellum, we can reach between 40 to 80% editing, so very efficient editing again in the cerebellum of SCA3 transgenic mice. And that's why we have both mouse and human genes which are analyzed, because the sequence we have targeted is conserved between mouse and human. So we can truly evaluate both wild-type and mutant Huntington expression. And as expected, we have a drastic loss of the expression of SCA3 in the cerebellum of these mice. So in the last experiment, we tested both the whole system together, so the ablate and replace with our AAV kamikaze, which is a self-inactivating system to get rid of the nuclease after a few weeks. And we obtain indeed a very nice inactivation in both cases. And as expected, we have the self-inactivation and the killing of the Cas9 in this animal to completely inactivate the nuclease activity after a while. And for the moment, we are pursuing the functional characterization of these samples. So I don't have other data to show you today. So to conclude, let me mention the people who did this work, and in particular, Sarah Reggio, Fabio Duarte, and Nicole Merienne for the Huntington work, and I thank you for your attention.
Thank you very much for a very nice talk. Any questions from the audience up there?
Very nice data. Thank you for showing this. In your single cell data, did you check whether your rescue correlates to the level of Huntington knockdown, or whether you, for example, need less rescue in endothelial cells compared to some others?
No, we didn't do that, but it's still quite complicated to do because we don't have yet the tool to do. I mean, the protein level, for example, it's in the total punch, and it's not at a cellular level. So we will need to go deeper and have truly multi-omic in single cells to ask the question you are asking. So we didn't do this type of experiment in this setting.
Another question? Yes?
Thank you for this excellent talk. I would like to, well, there are two obstacles to introduce this kind of therapy into humans. First of all, delivery to the brain and to a specific part of the brain, but previously very problematic was off-target effect, and I would like to ask you how did you resolve this problem in your lab, in your research?
Sure. So concerning the first part, the delivery, I think there is really, once again, a great revolution in the field and new delivery system, in particular vectors, which are emerging every day. And we have now more potent delivery systems which are available. And there are already in the brain several clinical trials which have demonstrated clearly that we can deliver in the brain and safely deliver in the brain. And there are clinical trials ongoing in that field. So I think the delivery, for me, is not really a limiting aspect. The second question is truly, indeed, a major concern. So that's actually why we developed this Kamikaze 9 system, because, I mean, once the gene editing, the CRISPR-Cas9 has cut his target, then it starts to do off-target. So that's why we have decided that we need to turn off the system. We could not afford the luxury of maintaining a nuclease in the brain forever. And so this Cas9 is self-inactivated, so after a few weeks, after the on-target is being done, then the nuclease is disappearing, the activity is disappearing, and we have shown in our paper of 2017 that by doing so, we reduce the off-target by, let's say, 80%. It's never black and white, it's improved, but they might still be off-target, and then it's truly the screening of the sequence that you use which is also making a big difference, like for ASO. I mean, you can take ASO, which have a lot of off-target, and others which are very good candidates. So it's also the screening of the candidate, which should make it more suitable for potential clinical application. And of course, we will need to do far more data to prove that it's safe and not inducing off-target, which might be undesirable.
Thank you.
Hey, thanks, Nicole, for this nice talk, as always. I have a related question more on the efficacy side. So you have quite limited effect in the cortex. Did you try different capsids to see if you can increase the effect?
No, we were quite conservative. We took, in that case, it was the AV10. No, sorry, the AV2 retro. And I would say, for the striatum, more than the capsid itself is first the site of injection in the striatum. So depending whether you are ventral, medium, dorsal, lateral, the projection to the cortex are not the same. So you need to cover really the whole striatum to be able to maximize what you see in the cortex. And then, of course, even if you, let's say, are great and you target 100% of the striatal cells, then in the cortex, depending on the region, now we know from the Atlas that you have between 15 and 20% or 25% of the neurons which project to the striatum. So in all cases, not all neurons in the cortex will be transduced, and all the other cell type will never be transduced. So we calculate, based on the Atlas and what we know of the connectivity, that at the maximum we can reach something like 25 to 30% editing in the cortex, and that would be, through this delivery method, the maximum we can reach. But these are the neurons which project to the striatum and might be highly relevant in HD. So maybe it's enough.
And what is your prediction for the larger brain? Maybe. Just a quick answer. Do you have
We will
an
need
idea?
to test with Romina.
Okay, thank you, thank you.
I will be happy to. Any more urgent questions? Well, if not, we're right on time. Thank you very much.
Sorry.
Il posera la pause.
And we can have a short break, right?
Yes.
Yes.
Thank you very
I can
much.
ask you a question in the break.
Merci, collègue.
Mais bien sûr, et injecter un CSF pour aller dans le cortex.
Oui, il faudrait avoir des bons... Oui.